the chemistry of metals in biological organisms. I'm going to be talking about the biologically important chemistry of metals. Why does nature use metals in biological systems? The answer is redox and coordination. Overall, the properties of metals that allow them to catalyze redox reactions and form coordination complexes is what makes them biologically important. When you think about metals, you might first think of solid bulk metals like gold, copper, iron, and silver. Or maybe you think about taking calcium supplements. You probably know that your blood contains iron and that your bones need calcium. You've also probably heard of lead poisoning and know that mercury is toxic. Today I'm going to be talking mainly about metals that are bound up in proteins and about transition metals. Other metals, as well as free metal ions, are biologically important, but outside of the scope of what I'm talking about in this lesson. Living organisms require varying amounts of metals. Iron, cobalt, copper, manganese, molybdenum, and zinc are required by humans, and approximately half of all proteins contain metal cofactors. Other metals, such as mercury, plutonium, and lead, are toxic. And by toxic, I mean that there aren't any vital or beneficial effects on organisms, and that the accumulation over time can cause serious illness. So why are some metals toxic and others vital? Why would our bodies even use metals? The answer, of course, is chemistry. The toxicology of metals is very complicated, another lesson in itself. However, generally toxic metals act in three different ways. They compete with and displace important metals. They have similar properties to important metals, and so can replace these metals in a binding site and disrupt biological processes. Toxic metals can also bioaccumulate. Our bodies don't have the ability to regulate or dispose of certain metals. The third way toxic metals act is through redox, by having a high oxidation state that our bodies can't handle. For example, chromium-3 is an essential trace element, but chromium-4 is a carcinogen. So let's talk about the properties of metals. They are shiny solids at room temperature, except for mercury, which is a liquid. They have large atomic radii, meaning that the distance from the nucleus to the valence shell is fairly large. They have low ionization energies. Ionization energy is the energy that is required to remove electrons from atoms. They have low electronegativities. Electronegativity is the measure of the tendency of an atom to attract electrons to itself. So what do these things tell us about metals? Do metals hold their electrons tightly or loosely? Do they more readily form cations or anions? The electrons are held loosely because the valence electrons are far away from the nucleus, so they can be readily lost. And losing electrons produces cations, so metals easily form cations. Because of these properties, metal ions are often cations and can exist in different oxidation states. Metal ions can be oxidized or reduced. They can lose or gain electrons. This is redox chemistry. When you hear the word redox, you probably first think about the electrolytic cells you learned about in Gen Chem, where the redox potential, the tendency to be reduced or oxidized, drove a chemical reaction that produced energy as a current. You've also probably seen half reactions, like this. Now let's consider some of the redox chemistry that is going on in organisms. Our bodies break down glucose through oxidation to produce energy. In this process, metals can act as terminal electron acceptors. This schematic shows the oxidation of glucose into different molecules, which feed into the Krebs cycle, or the citric acid cycle. And electrons are lost along the way, which reduce NAD+ to NADH. which goes through the electron transport chain, and among other species, iron 3 is reduced to iron 2. So iron 3 is the terminal electron acceptor at the end of this process of oxidizing glucose. Another example of an important redox reaction is the enzymatic superoxide dismutase reaction. You've probably heard that free radicals are bad for you, and so you should buy lots and lots of pomegranate juice because it's full of antioxidants, right? Well, maybe, but your body has its own way of getting rid of free radicals. Through redox reactions, the oxygen radical superoxide can be transformed into dioxygen or hydrogen peroxide. Superoxide dismutase is the enzyme that does this with the help of a metal covector. Different types of SOD exist that can use copper or zinc, manganese, iron, or nickel. The metal is bound up in the superoxide dismutase enzyme and is the important atom in the active site. The reactions and half reactions are the following. You can see from the reaction where hydrogen peroxide is produced that the metal is being oxidized and that the oxygen radical is being reduced. In the second set of reactions, where dioxygen is produced, you can see that the metal is being reduced and that the oxygen radical is being oxidized. The metal ion in superoxide dismutase is bound up in the enzyme by coordinating with the amino acids in the enzyme. This brings us to the second category of metal chemistry, coordination. Coordination chemistry involves transition metals. As you might recall, transition metals are defined as elements that have incomplete d orbitals or can form cations that have incomplete d orbitals. So the tendency of metals to form cations also allows them to form coordination complexes. The metal ion acts as a Lewis acid, accepting a pair of electrons. The donor atom, or molecule, called the ligand, acts as a Lewis base. The donor atom must have a free lone pair of electrons to donate. The metal and the ligand form a coordinate covalent bond. Common donor atoms include oxygen, nitrogen, and sulfur. Examples of ligands include water, cyanide, oxygen, halogens, ammonia, etc. These are all monodentate ligands. But there are ligands with more than one donor atom that can coordinate to the metal. These are called polydentate ligands. You might notice that dentate has the same root as dentist, and I like to think about the ligands biting down on the metal. Polydentate ligands bite down more than once on the metal. Monodentate ligands only bite down once. Polydentate ligands are also called chelators and are said to chelate the metal ions. You've probably seen models or pictures of coordination complexes that look like this. And you know that they have different geometries based on the number of ligands and the number of atoms coordinating to the metal. The number of atoms coordinating to the metal is the coordination number. So for example, octahedral complexes have a coordination number of 6. You may have learned how to name these compounds and heard about crystal field theory and how the d orbitals are split in energy. For example, in an octahedral complex, the dz squared and dx squared y squared orbitals are higher in energy because those orbitals lie in the same plane as the ligands and therefore have repulsions. The other orbitals don't lie in the same plane as the ligands, and so those orbitals, the dxy, dyz, and dxz orbitals, are lower in energy. You probably learned that this splitting energy and the dd transition of electrons leads to colored solutions. But the complexes you saw probably only had simple ligands and seemed unrelated to biological function. So how does this relate to the chemistry of metals in organisms, besides the fact that our blood is red, absorbing green, and that horseshoe crabs have blue blood, meaning that the delta corresponds to orange? Earlier, I talked about metalloproteins, and we've already reviewed that donor atoms are commonly sulfur, nitrogen, and oxygen. So what are the building blocks of proteins? Amino acids. And amino acids can contain sulfur, oxygen, or nitrogen. Metal ions are part of proteins through coordinating to amino acids. In this way, metal ions can give shape to proteins and are often found in the active site of enzymes, where the chemistry is done. Histidine, cysteine, and serine are just examples of amino acids that can coordinate to metals. Histidine with a nitrogen donor atom, cysteine with sulfur, and serine with oxygen. You can imagine that any amino acid with an atom that can donate a pair of electrons could coordinate to metals. An example of amino acid metal coordination is the zinc finger. Zinc fingers are structural motifs that are commonly found in proteins. Zinc coordinates to histidine and cysteine and forms the zinc finger structure. Structure often plays an important role in the function of a protein. Zinc fingers typically function in binding DNA. Coordination chemistry is also involved in the transportation of molecules. For example, the transportation of oxygen in blood. Hemoglobin is an iron-containing metalloprotein that transports oxygen. As you can see, iron 2 coordinates with heme and then can reversibly coordinate to oxygen. On the left, you see the deoxygenated iron coordinating to heme and then coordinating to oxygen as well. So why metals? Hopefully that question has been answered. The properties of metals, namely that their electrons are held loosely, they can exist in different oxidation states and therefore readily perform redox and coordination chemistry. The chemistry of metals makes them indispensable to organisms.